Hi, I'm Sanhita Sinharay. I'm an assistant professor in the Department of Bioengineering at the Indian Institute of Science, Bangalore. To give a short summary of my research journey so far, I did my bachelor's from Jadavpur University and my master's from IIT Madras, both in chemistry, and then I did a PhD from University of Arizona in Tucson, Department of Chemistry and Biochemistry. And after that, I did a postdoctoral fellowship at the National Institutes of Health in the Department of Radiology and Imaging Sciences Clinical Center. Before joining IISc, I was a research instructor at MD Anderson Cancer Center in the Division of Cancer Systems Imaging. My journey has been very multidisciplinary, and I was trained as an organic chemist, trying to develop MRI contrast agents to specifically look at physiological biomarkers of the tumor microenvironment in a non-invasive manner. I focused on chemical exchange saturation transfer, or CEST MRI contrast agents, for my PhD. And I was very excited actually about the prospect of translation of these contrast agents in the field of biomedical engineering, and to see the application of the chemistry side of things, taking it from the bench to the bedside by translating them within preclinical animal models in a small animal MRI scanner. I shifted gears a little bit during my postdoctoral work, where I moved from cancer imaging towards infectious diseases imaging, mainly focusing on understanding the neuropathology with non-invasive PET, which is positron emission tomography imaging, or MRI.
At IISc, our molecular imaging research team again combines multiple disciplines towards molecular imaging, biomarker discovery and translation. Primarily, our group focuses on magnetic resonance imaging, magnetic resonance spectroscopy and photoacoustic imaging as the main study modalities. And leveraging these imaging methods, we aspire to non-invasively image the tissue microenvironment, specific biomarkers in the tissue microenvironment such as pH, enzyme upregulation, metabolites, specific receptors, thereby bringing about translation and in the field of molecular diagnostics. A major part of our team also focuses on developing and validating contrast agents within preclinical models. The purpose of development of the contrast agent is to significantly enhance the specificity of lesion detection, aid in image-guided surgeries and assessment of therapeutic response, for example, with the agents that we are developing as theranostics. Our molecular imaging research team has a mix of people from various disciplines as well. We have chemists, biologists, engineers, as well as clinicians in our team. And we look forward to solving biomedical challenges through the interplay of all these interdisciplinary concepts and skills.
Hi, I am Ananya Sharma, a fifth-year PhD student in Molecular Imaging Research Laboratory, working with Dr. Sanhita Sinharay.
I'm always excited about my research and would be happy to share that here. I work on photoacoustic imaging, primarily focusing on leveraging different chemical and physical properties of dyes to develop novel photoacoustic based contrast. In this context, I have developed modified zinc phthalocyanine based photoacoustic contrast agents that have shown excellent theranostic abilities in hypoxic pancreatic tumor models. Further, I have ventured into the realm of biomarker targeted contrast agents and their development and translation towards tumor specific diagnostics.
Hi, I'm Ankita. I'm a second year PhD student. I joined IISc in 2024 and I'm working in molecular imaging research lab with Dr. Sanhita Sinharay.
I completed BSMS from IISr Kolkata in biological science, and from a core biological background, I have joined in an interdisciplinary field. In this lab, I am experiencing how chemistry and biology is being studied parallelly, and through this also I get to know how chemistry is helping towards clinical application. I am working on targeted diagnosis of triple negative breast cancer or TNBC through photoacoustic imaging. TNBC is a subtype of breast cancer that doesn't have three hormonal receptors, that is estrogen receptor, progesterone receptor and HER2, which normally other breast cancer types express. Despite lacking these receptors, they can grow relying on alternative growth factors, signaling pathways, and can become more aggressive and early metastatic. Due to absence of these molecular targets, conventional imaging techniques and treatment approaches are often insufficient for early detection and precise diagnosis. By using targeted photoacoustic probes that specifically go and bind to TNBC biomarkers, this approach might enable sensitive and non-invasive detection of TNBC.